Okay, so here we're going to talk about transcription in hopefully what's a short video. So when we talk about the purpose of transcription, transcription is basically making a copy of a gene that we find in our DNA. Now, the reason we make messenger RNA is to carry that directions on how to build a protein to a ribosome. Now, the ribosome in a eukaryote could be attached to the rough ER or the cytoplasm. So the messenger RNA is a disposable copy of the gene, whereas the DNA needs to stay intact within the nucleus. So our main enzyme that we use in transcription is called Rna Polymerase. Now, Rna Polymerase is going to unwind the double helix, and one strand will be used from the DNA. So that strand that is used is called the non-coding strand or the template strand. And so here, it's important to know that messenger RNA is built from 5 to 3, same as DNA. And it's going to run anti-parallel, the same as 2 strands of DNA. So therefore, we're going to pick the strand of RNA that is opposite of 5 to 3. So in this picture here, that's going to be the top strand. So the top strand here is going to be the template strand that we use, and we're going to follow base pairing rules. Now the difference, what we've seen so far, G pairs with C, awesome. But now you see that there's an A in DNA. Normally we would think a T pairs, but there are no T's in RNA. Instead we have uracil. Now you also notice a different color in the sugars. So DNA has deoxyribose, whereas RNA has ribose. Okay, so now we'll have G, and then U, and then G, and then A. Oops. So in that purple nucleotide, it'll be A next. Now, as RNA polymerase moves down the gene, the RNA transcript that is being built will start to tail off. So you can see how the double helix is going to zip back together, and the RNA begins to trail off, and then right there. So then RNA polymerase will continue to do base pairing rules, building a single-stranded messenger RNA molecule. You can see it's being built in the 5' to 3' direction. So on paper, this is what it would look like. If you have one strand of the DNA and you're asked to do transcription or find the mRNA sequence, so here our first codon or our first bases, we would have T pairs with A, and then A pairs with U, C goes with G. Then our next one, we have G pairs with C, C pairs with G, T pairs with A. Now be careful here. A lot of times kids get a little hesitant or nervous. So if you see a T in DNA, that's okay. It's supposed to be there. And there are A's in RNA. However, you notice in this next codon, there is a T. Well, so then it pairs with an A. But now the A pairs with a U. That's what's different. Anytime you're thinking of writing a T in RNA, switch it to a U for uracil. Okay? So now that is transcription and what it looks like on paper. And then RNA polymerase will eventually finish transcribing the gene, and then you have your RNA transcript. All right. Now in bacteria, this would be it. This would be the RNA, and ribosomes would already be attaching and translating. Now in eukaryotes, the next step is RNA processing. All right, great job.